Hello everyone, and welcome to today's webinar e-Congress International COVID-19. My name is Rosalino Vázquez López, and I am a researcher at the Faculty of Health Science of the Universidad Anáhuac, Mexico. One of my main lines of research is focused on the study of intracellular signaling pathways in the cell death by apoptosis. Likewise, my team and I are currently studying the pathophysiology of acute respiratory distress syndrome. It is a pleasure for me to introduce this presentation on behalf of my co-authors, Alfonso Ceniceros, Alonso Asensio, Dr. Diego Álvarez, Master Science Sandra Solano, and Dr. Claudia Ferreira. Next, Alfonso Ceniceros, a member of my research team, will present the structure of alveolus and the relationship with the acute respiratory distress syndrome in the COVID-19 context. Welcome, Alfonso. Please share your presentation. Thank you.
Hello, everyone. I am Alfonso Ceniceros Obregón. I want to thank Rosalino for introducing me and all my teammates for this work. I'm going to present you the cellular and molecular events that occur in the alveoli in ARDS in a specific focus on COVID-19. Let us have to look to the background of the presentation. The acute respiratory distress syndrome, or ARDS, is a clinical syndrome that has a considerable number of morbidity and mortality numbers. Although there are different types of conditions that can cause ARDS, the majority of reported cases are from pneumonia as well as sepsis and a small number of different diseases, including COVID-19. Patients that survive ARDS constantly seem affected by different long-term sequelae, as well as 3-year mortality rate. Acute respiratory distress syndrome has been reported that clinically is very similar to other conditions that can cause very similar symptomatology, such as acute heart failure and alveolar hemorrhage. ARDS begins with a tisular injury and almost destroys the alveolar microvascular architecture because of the inflammatory injury of the tissue. Our objective is to describe the acute respiratory distress syndrome and its phases to study it and be able to distinguish it from other signs and symptoms. Our methodology was a systematic analysis of documentary research of the structure of the alveolus and its relationship with an ARDS, with a focus on COVID-19. Acute respiratory distress syndrome pathophysiology. First of all, I want to talk to you about the structure of the alveoli. The alveoli has an hexagon structure that we can see in the image. It has the AT2 and the AT1. This is the alveolar cell type 1, and this is the alveolar cell type 2. The alveolar cell type 1 moves through the alveoli towards the outlet of the respiratory system, and the alveolar cell type 2 generates the surfactant factor that protects the alveoli of collapsing. We can see here the NAC and the sodium-potassium ATPase. And in the interalveolar septum, we can see the fibroblasts, the mast cells, the lymphocyte, the monocyte, and the collagen type 1. Here are the lymph vessels. There are various pathologies that can be considered as primary diseases that trigger the condition of acute respiratory distress syndrome. For example, COVID-19, pneumonia, aspiration, trauma, among others. ARDS begins with a primary disease which produces tissue damage and destruction of the alveolar microvascular architecture. Inflammatory damage begins with the cytotoxic attack mediated by cells of the immune system as neutrophils, macrophages, and platelets. It has been described the ARDS can occur in different phases: in the exudative phase, proliferative phase, fibrotic phase, and the repair phase. In the exudative phase, has some specific characteristics. It begins with the alveolar edema caused by the permeability in the alveolar capillary in the presence of neutrophils and erythrocytes, creating an exudative state that is very important in this syndrome and inflammatory process. The inflammatory process produces a lysis of the alveolar epithelium, principally of the alveolar cell type 1 and the alveolar cell type 2. Could almost activate the alveolar macrophages and activating them in the NFKB pathway against infected agents. These events caused by the neutrophils can be grown an inflammatory response in the alveolar tissue. Loss of the endothelial and epithelial cell causes the vascular endothelial growth factor known as VEGF decreases because the VEGF is synthesized and released by the vascular endothelial lung epithelium. Another characteristic of this phase is that there is a development of highly membranous alveolar increases in interleukin 8 and TGF-beta, and the inhibition of the INA-C sodium key ATPase. As we can see in the image, there is the injured epithelium and injured endothelium here. The INA-C K ATPase inhibited. The exudative within the alveolus, that here is the different type of cells inside the alveolus, like the platelets, macrophages, neutrophil, erythrocytes, there is somewhere. And the different analysis of the 81 and the 82, we can see here. Okay, and the hyaline members here. Proliferative phase. In this phase, there is a presence of exudate and hyaline membranes, alveolar hemorrhage, fibrin deposition, and in some areas of the alveolar atelectasis, that is the collapse of the alveolus. May also occur, these events exacerbate the low alveolar capacity. Also, it has very important characteristics such as formation of hyaline membranes, as I said, alveolar cell infiltrate, because although the alveolar exudate initially inhibits the function of the surfactant, and as the epithelial damage progresses, it comes to inhibit its synthesis as the exudate spreads and the surfactant loses its function. Alveolar permeability and the function of epithelial ion channels are also lost. Weaving will lead to the alteration and losses of the osmotic forces responsible for preventing the arrival of edema to the alveolus and returning to the interstitium. PEGFR1 and 2 increases, and the neutrophil produces ROS, that is a reactive oxygen species, and normally, as a restoration mechanism, it will start restoring the number of 82 followed by the 81. This process is called transfermentation. In the image, we can see the INA-C reactivation as the sodium-potassium ATPase reactivation, the edema removal via lymphatic, and the transfermentation process, okay, with the 82 proliferation, and the ion channel. In the fibrotic phase, we can find the excess of vascular damage, vascular fibrosis, severe progressive hypoxia, and sepsis that commonly leads the patient to death. In the image, it's important to notice the fibroblasts in the alveolus and the capillary fibrosis. If the patient improves, we will enter to the recovery phase or the repair phase. And it's important to emphasize that in this phase, you may or may not regain normal lung capacity. As a conclusion, The emergence of the COVID-19 disease has represented great challenges for scientific research, from the clinical management of the patient to the need for the development of future specific therapeutic schemes and the development of vaccines. In this context, the understanding of the molecular, cellular, and tissue banks of the pathophysiology of the disease play an extremely important role. At this time, we are at the beginning of the decipherment of the many of the processes involved, which, little by little, are shedding light to understand more about the pathophysiology of the disease. Thank you all for listening. I hope this presentation has been to your liking. And if someone would like to know about the references used in this work, please scan this QR. You can use your smartphone. Thank you and goodbye. Alfonso Cenicero.